Rarely get that. Everybody had a good weekend? Oops. And I ruined it? How did I ruin your weekend?
You gave us our test back on Friday.
That's supposed to be good news. No. Well, okay, I'm sorry. You didn't have to pick it up, okay? Nobody said, go pick up your exam, right?
Is the curve shown now or at the end of the course?
The curve is, it's right here on the web page. If you look right there, there you be. That shut everybody up. So it's on the web. It's on the web page. You can see it. Okay. So I've got a fair amount to get through today, and I probably won't make it all the way through, but that's fine too. I want to continue our discussion that I started last time about mechanisms of controlling enzymes. And if you recall, I finished just about the place where I was talking about a model of control. So when I talked about hemoglobin, I talked about how binding of the first oxygen affected the binding of the second, affected binding of the third, affected binding of the fourth. And I said that this was a sequential model. That is, that each one influenced the one after it. And that's one way of explaining how it is that cooperativity that we see in hemoglobin exists. When we talk about enzymes, of course, we don't talk about cooperativity so much as we talk about control. And there are other models that are used to explain that. And I want to explain a couple of those models to you. The first of these models is called the concerted model. Okay? And the concerted model is a little odd, at least to understand. Let me back up. It's a little odd to understand to begin with because it has a different setup than the sequential model. So the sequential model looks like what you see on the screen. Okay? The sequential model said that we started out with a protein, maybe it was hemoglobin or maybe it was an enzyme, and that protein was in one state. We can think of this as being in the T state. And binding of the one molecule to one subunit caused it to affect the other subunit. So you see how this guy has bound a substrate, and you can see how these other subunits next to it have changed from being squared off to being rounded off. That is, their structure has been affected by the binding of the first one to the very first subunit. We can think of this as a cause and effect. This causes these to change, this causes these to change, this causes this to change, et cetera, et cetera. So we see a cause and effect. Binding of one causes the next subunit to change, causes the next subunit to change, et cetera, et cetera. All right? So that sequential model is sort of an intuitive one that we think about, but there are other ways of explaining cooperativity in hemoglobin and also control in enzymes. The second one that I want to talk about is called the concerted model. And the concerted model, I don't have a good one to show you here visually, unfortunately, but I'll have to describe it to you. The concerted model says, let's go back and look here. All right. The concerted model says there's not a cause and effect. There's not a cause and effect. The enzyme can exist in two states. It can exist in a T state, and it can exist in an R state. And the flipping from T to R has nothing to do with the binding of the substrate. You think, well, how does it do it then? It has nothing to do with the binding of the substrate. It has nothing to do with the binding of the allosteric effector. What the concerted model says is the flipping is an inherent activity of the enzyme. The enzyme is capable of flipping from R to T completely independent of another molecule. The sequential model says it was a cause and effect. The concerted model says it's flipping independent of that. Well, then how do we see the phenomenon exhibit itself? What the concerted model says is that after the flipping occurs, then something can bind, and that something that binds locks it into one state versus the other state. So if I have a an enzyme that flips into the R state, and if this enzyme is ATCase, and this enzyme binds to ATP, it locks it in the R state. Therefore, that enzyme will remain in the R state and be very active. If the enzyme lets go of ATP and it flips back to the T state all on its own, and it binds to CTP, it locks it in the T state. And it stays locked in that state until it lets go of CTP. So these are fundamentally different ways of explaining R state versus T state. In the sequential model, the R to T flip occurs as a result of binding. In the concerted model, the flipping is independent of binding, but binding locks it into one or the other. Okay? Now, I know that's always a little conceptually difficult to get your heads around, so I'll stop and take a brief question for that. Yeah?
Is one or the other more accurate, or is it a hybrid of the two?
Is one or the other more accurate, or is it a hybrid of the two? It varies from one protein to another in terms of which one it tends to be more like. So there are some that will be very much more on the concerted side, others that will be very much more on the sequential side. And all I'm expecting you to do is not memorize which one is which, but to just understand what the two different models tell us. Yes?
Is there something that causes it to unlock them later, or is it time-driven, or...
Is there something that you say unlock? Do you mean to let go of the molecule? Okay, so there's something that causes it to let go of CTP, or something that causes it to let go of ATP or aspartate, right? The answer is, like every other binding we've talked about that's not covalent, these are associative things. They come on, they go off. They come on, they go off. So we always see reversible binding of these intermediates. If they're covalently bound, then of course we don't have any way of getting them off of there. Yes, sir?
Would the state change be spontaneous of itself, or would it result from a reaction of something other than the independent substrate, like the
Yeah. Good question. So his question is the change R to T completely independent of everything else? And the answer is yes, it is. Yes, it is. And we will see preferences for one versus the other. I told you that when we started studying ATCase, we didn't even really know the R state existed until we locked it with Pala, right? And that's because that the preference was something like a couple hundred to 1 of T over R when there's nothing there. Okay? A couple hundred to 1 when there's nothing else there. But if we lock one of them in the R state, then that equilibrium starts moving in the direction of the R state. More and more start flipping into the R. Yes, sir?
Does this rate of T to R transition switch, is there any trend to where something that switches states spontaneously very quickly favors a concerted model?
I'm not sure I understand the question.
If an enzyme spontaneously changes states very rapidly
Yes.
relative to other enzymes, would that be better described by the concerted model, since there would be more opportunity for
Okay.
change volume?
So his question has to do with how rapidly the T to R state change occurs within an enzyme, and does this tend to favor one model versus the other? The answer is, if it's independently changing, it already is in the concerted model. So if the changing is independent of a substrate binding or an allosteric effector binding, then it automatically is a concerted model. Okay? Okay, so I think that you should, as I say, understand the generalities of those models and be able to explain them to me in words. That would be useful. I want to turn our attention now from talking about models, but to talking about some interesting enzymes. And this first enzyme I'm going to talk about is one you're going to hear a lot about. Yeah, question?
Oh, so that graph was not so important that you kind of, for the concerted model graph?
So you're talking about here?
Yeah.
Yeah. So all this graph is showing is, okay, so here is the ratio of T to R of 70. Okay, this is when ATP is present. Here's the ratio of T to R, that's 200 when there's nothing present. And here's the ratio of 1250 to 1 when there's CTP present. This is consistent with what I've told you before. That is, ATP favors the R state more. So we see less of a ratio. That is, less of a ratio. L is the ratio of T to R. Okay? Nothing is 200. And when we have the inhibitor, in this case CTP, it's 1250 to 1. Okay? Okay. Now, As I said, I want to talk a little bit about an enzyme. You're going to hear a lot more about this term. We're going to see that this enzyme plays a very, very important role in a phenomenon we refer to as signaling. Signaling is a way for cells to talk to each other. And for one cell to talk to another, it must release something like a hormone. That hormone goes to that other cell and binds to that other cell and causes changes to happen inside the target cell. And one of the enzymes that plays a role in those changes in the target cell is protein kinase A. You're going to hear a lot about protein kinase A. So it's important that we understand, first of all, how it works. Protein kinase A is allosterically affected by a molecule called cyclic AMP, or as we will refer to it in this class, CAMP. You see it right here. The structure of CAMP is shown here. No, you don't need to know the structure. It's related to AMP, AMP being adenosine monophosphate. The cyclic part is this little ring out here. Okay? So AMP doesn't have a ring, it just has a phosphate hanging out there, but when we make it cyclic, we actually rejoin it to this and make a circular structure. Cyclic AMP turns out to be very, very important for this communication process that cells go through. Very important. And the reason it's very important is because cyclic AMP affects protein kinase A. As I said, it is an allosteric effector of the enzyme. And the way it allosterically affects the enzyme is different than we've talked about other allosteric effectors. So far with allosteric effectors, we've talked about the T to R or the R to T conversion. We've seen shape changes that occurred as a result of binding of allosteric effectors. Cyclic AMP is an allosteric effector, but it's not doing T to R changes. It's doing something like shown here. It turns out protein kinase A has multiple subunits, like you've seen before in other enzymes. However, this guy has catalytic and regulatory subunits, like you saw with ATCase, but in this case, the binding of cyclic AMP causes something different to happen. The something different that happens is binding of cyclic AMP causes the catalytic subunit to let go of the regulatory subunits. This is literally an on-off switch. Remember I said that with T and R, we don't really have on-off switches. I said we turn the volume down or we turn the volume up. This is an on-off switch. When the regulatory subunits are bound to the catalytic subunits, as we see on the left over here, this guy's dead in the water. It's not catalyzing anything. I'll tell you what it catalyzes in a second. Okay? When cyclic AMP is present, it binds to the regulatory subunits, releases them from the catalytic, and these catalytic subunits are active as all get out. Okay? So cyclic AMP is literally turning the enzyme from the off position to the on position. How do we turn it back off? Well, if we start degrading cyclic AMP in the cell, when this guy comes off, it'll get degraded. And remember, this is a reversible binding again. These are coming on, coming off, coming on, coming off. As we start degrading cyclic AMP in the cell, these guys will move back to this state, and they will grab a hold of the catalytic subunits and turn them off. Okay. Now, these guys work by literally blocking the active site of protein kinase A. They block the active site. They physically provide a barrier. The active site can't get at what it normally catalyzes, and as a result, is dead in the water. It doesn't do anything. What does protein kinase A catalyze? Okay? Well, first of all, I need to tell you a term that you should absolutely know, and that's the term kinase. First of all, whenever you hear the name of a molecule or the name of a protein in this class ending in ASE, it is always an enzyme. Always. So the fact that kinase ends in ASE tells us that it's an enzyme. Kinase is an enzyme that puts phosphates onto molecules. It puts phosphates onto molecules. So when you hear the term phosphate, it's putting a phosphate onto something. The name protein kinase tells us that this enzyme puts phosphates onto other proteins. So protein kinase A, that just happens to be the very first one, protein kinase A catalyzes the addition of phosphates to target proteins. Now, on these target proteins, the effects can be enormous, as we shall see. But for the moment, all you need to know is that protein kinase A, when it's active, is putting phosphates onto target proteins. Okay? Everybody got that? So kinase puts a phosphate onto target proteins. There have to be enzymes that take phosphates off, and I'll talk about those in just a few minutes. But a kinase puts it on, a protein kinase puts it onto other proteins. And as we will see in this signaling process that I have described to you, addition of a phosphate to a protein can make a protein be much more active or much less active. So now we start to see another level of control of enzymes. Allosteric control was one. And we've seen a couple of examples in the form of ATCase and also protein kinase A. Protein kinase A covalently modifies targets, those targets being other proteins. And that covalent modification is a regulatory mechanism as well. Okay? Covalent modification in the cell occurs in many ways. Putting a phosphate on or taking a phosphate off is one covalent modification. There are other covalent modifications, and you've already seen one example of those. When I talked about the proteases, Chymotrypsin is covalently modifying its target because it's cutting it in half. It's breaking a covalent bond. Okay? These are different modifications that can occur inside of a cell. Phosphorylation, acetylation, meaning putting on an acetyl group, myristoylation, putting on myristic acid, blah, blah, blah. Okay? I'm not giving you this table to give you something to memorize, but I do want you to be aware that there's many different kinds of modification to proteins that can occur. Those modifications affect the proteins in significant ways because frequently they will change the charge of the protein. If we put a phosphate onto one section of a protein where there wasn't a charge before, now all of a sudden it might be attracted to another part of the protein that's positively charged. It might be repelling a part that's negatively charged. And so we could see this then as a mechanism of changing protein shape. And changing protein shape changes protein function. Okay. Now, let's talk about protein kinase. As I said, protein kinase, or protein kinases, specifically protein kinase A, takes a phosphate and it puts it onto a target protein. Where does it put it on? Primarily, it puts it on to the amino acids that contain hydroxyl groups. These include serine, threonine, and tyrosine. And when we look at the enzymes that catalyze these, we see that they can be grouped into different categories. Some protein kinases strongly prefer or absolutely prefer to put them onto serine or threonine. Protein kinase A is one of those enzymes. It strongly prefers to put a phosphate onto either serine or threonine side chains. Other enzymes, other protein kinases, prefer to put it onto tyrosine. And we'll see when we talk about signaling that the tyrosine kinases are really interesting. They're intimately involved in controlling cells' decision to divide or not divide. This plays a role in cancer and many other processes. Okay? I'm sorry?
Protein kinase A prefers serine and threonine.
Protein kinase A works on serine and threonine. Okay, so that's the reactions being catalyzed. We see that ATP is the source of the phosphate. Here's our target hydroxyl group. Here's the phosphate that's put onto it. That leaves us with ADP as a byproduct. Putting phosphates on is one thing. Taking phosphates off is another. Okay?
Is ATP always the source of that phosphate?
Is ATP always the source of the phosphate? Never say always, but mostly I would say yes. Yes. Okay? Removing phosphates is a phenomenon known as dephosphorylation. And it's not the reversal of a phosphorylation. Instead, separate enzymes are used. And the enzymes that remove phosphates off of molecules or proteins are known as phosphatases. We'll talk a lot about this enzyme called protein phosphatase because it reverses the action of protein kinases. It catalyzes the hydrolysis, that is the use of water, to split off a phosphate off of a target. So here's the phosphorylated protein that we had from the protein kinase. If we treat that protein with protein phosphatase, we see that we get phosphate and the protein back in its original state. This is important because, as I've said before, cells are control freaks. If they have a way of turning something on, they darn sure better have a way of turning it off, just like you better have a way to turn off your lights or your electric bill is going to go crazy, right? So too, cells want to turn off a process or they may burn up all their resources. So if they have the ability to put the phosphate on, they darn sure better have the ability to take the phosphate off, and that's what we see in the form of protein phosphatase. Understanding signals that cells are getting requires us to understand which of these enzymes is active at any given time. And that's a fascinating process. Okay, I'll stop, slow down, and take questions. Clear as mud? Am I going slow enough for you? What's that? Clear as mud. Oh, you like that saying. Okay, well, that's good. All right. So, phosphorylation is important. Dephosphorylation is important. Let's now think of some other covalent modifications. Some other covalent modifications. Okay? So I've already alluded to the fact that breaking peptide bonds is a covalent modification. And this covalent modification turns out to have some really cool implications for human health. Very, very cool implications. They're involved in digestive processes and controlling the enzymes of digestive processes. They're involved in blood clotting. And I'll be talking about both of these. The first one I'll talk about is digestive processes. If we think about it for a moment, we sort of take digestion for granted until we get an upset stomach, right? We get an upset stomach and then we become very aware of digestion. But in the absence of that, we eat our turkey and we go to bed and we fall asleep and that's that. And we wake up in the morning and we haven't given a thought to digesting something, right? But what's been happening after we eat is that we have digestive enzymes that are breaking down those things that we've been stuffing down our gullet the whole day. With Thanksgiving coming up, I have turkey on the mind, you can see. Okay? Now, it's important that the body control those digestive enzymes. I don't want those digestive enzymes digesting me. And those digestive enzymes are really, really good at digesting things. Okay? Now, when we look at the digestive enzymes, they're made in a variety of places, but one of the more common places that they're made is by our pancreas. Our pancreas makes digestive enzymes. And these digestive enzymes come in a variety of categories, but we're going to focus for the moment on ones that break down proteins. Okay? These are proteases. We eat meat. We break the protein in meat down to amino acids so we can use those amino acids to make our own proteins. All right? I don't want my digestive enzymes to digest my pancreas. Right? I'm fond of my pancreas. All right? So my pancreas is not stupid. It doesn't make active enzymes. It makes inactive enzymes called zymogens. So in other words, it starts out making these enzymes that will ultimately become active, but it starts out making them in an inactive form. So it can get it away from itself without digesting itself in the process of being made. So these inactive enzymes are known as zymogens. We can see them being made by the pancreas, and they're actually secreted outside of the cell, so they can go into our digestive tract where they get activated and start breaking things down. You might say, well, don't they sort of digest the digestive tract itself? And the answer is, to a limited extent, they do. Some of the cells that have the most rapid turnover in our body are those found in our digestive tract. Okay? When you hear of ulcers and people getting ulcers, what's happening is they're not replacing those cells as rapidly as they're being degraded. You have damage that's happening as a result of that. Yes, sir?
Aren't those primarily attributed now to Helicobacter pylori?
So his question has to do with ulcers relative to Helicobacter pylori, and the answer is yes, but that plays a role, again, also in this turnover of cells. So the turnover of cells is a very important consideration in the digestive tract. We used to say it was stress that caused that phenomenon. Now we know there's other factors that play a role in that. Okay? Now, what happens if we release our digestive enzymes from our pancreas, and that activation process, which I'll show you about in a second, starts backing up the tubes and getting up to the pancreas? Will that cause a problem? Oh, yeah. Oh, yeah. You create a condition that's very painful, in some cases fatal, called pancreatitis. And what's happening is those zymogens are getting activated in the wrong place at the wrong time. The pancreas is getting chewed up. It's treatable, but it's a very painful condition. Has anybody here ever had pancreatitis? Usually have one or two that have. Nobody that'll raise their hand. Okay? It can be life-threatening in some cases. It's a very painful condition, and it is treatable, but it has to be treated fairly early. Okay, well, how do these things get activated? Well, it turns out that here's one example. It shows how our body activates the enzyme you learned about last week, known as chymotrypsin. Chymotrypsin is made in an inactive form, and the inactive form is known as chymotrypsinogen. Whenever we see the suffix ogen on the end of it, we know we have a zymogen, and a zymogen is inactive. Unfortunately, that ogen is not consistent. We see that that was used after they named some other enzymes, and I'll show you those in a second. But for the moment, we have chymotrypsinogen. Here's how it's made. It's made as a 245 amino acid piece, and it's inactive. It doesn't do anything. It goes off into the digestive system where it encounters proteases that start to chew it up. And that starting to chew it up actually activates it. Okay? The first thing that happens is an enzyme known as trypsin makes a single cut between amino acids 15 and 16. And no, you don't need to memorize these numbers or anything like that. This creates something called pi-chymotrypsin that is partly active, meaning that pi-chymotrypsin will cut other pi-chymotrypsins. They're sort of a self-digesting set of enzymes. Pi-chymotrypsin cutting pi-chymotrypsins causes other bonds to be broken. We see that the bond between position 13 and 14 got broken. So now we lose a couple of amino acids there. We see that the bond between 146 and 147, and also between 148 and 149 got broken. And now chymotrypsin's in 3 pieces. Why don't these 3 pieces go flying away? Any thoughts? Sir?
Are there disulfide bonds present?
There are disulfide bonds, absolutely. So this enzyme has to fold properly. The disulfide bonds have to form, and then the cleavage occurs. Because if that doesn't happen, then these pieces all go flying away, and we have no active enzyme at all. It's prime evidence for the process of folding. It's prime evidence for the importance of disulfide bonds. Now, after we've gotten this pi-chymotrypsin making these various cleavages, this guy down here is what we call chymotrypsin. It's completely active. And now it can go out and start breaking down those things that you've been eating over Thanksgiving. Okay? What's happening in this process is we're looking at the inactive form in red, we're looking at the active form in blue, and for the most part the structure doesn't change much, but we look at what happens as a result of this cleavage. Look where this amino acid number 16 is up here, and look how far it gets moved after that cleavage has occurred. Why is that important? Well, what this is doing is it's opening up access to the active site. The movement of this amino acid, this distance, allows substrate to now come into the active site and be cleaved. Okay? Shape changes, essential for protein function. This shape change happens as a result of proteolytic cleavage.
So those three pieces do stay linked because of the disulfide bonds.
The three pieces stay linked because of their disulfide bonds. That's correct. Mm-hmm. Okay. So that's pretty cool. How am I doing on time? Okay, we're doing very well. Now, I got to tell you a cool story. Well, actually, before I tell you the story, let me show you also the bigger process. I've shown you the activation of one of these guys. It turns out that a bunch of the proteolytic enzymes in our body, as well as other enzymes in our body, in the digestive system, are all synthesized as in the inactive form. Okay? Trypsin itself is synthesized as trypsinogen. Okay? Here's chymotrypsinogen that you saw here. Here's procarboxypeptidase. That's another enzyme that breaks down proteins. Here's proelastase. Generally, when you see a pro on the front, that's the equivalent of an ogen on the end. It means it got named before they started using ogen to name them. Proelastase gets converted to elastase. And I'm going to talk more about that one in just a minute. There's a master switch on this scheme, and that master switch comes from this enzyme known as enteropeptidase. And no, I'm not going to tell you how enteropeptidase gets activated. Okay? We can imagine we could follow the scheme back quite a ways, and we can. The control of this system is important. If we lose control of this system, we end up making things like pancreatitis. We have enzymes active where we don't want them to be active. And that's not a good thing. Okay? Notice that we have over here prolipase going to lipase. A lipase is something that breaks down fat. We're also controlling enzymes that break down fat. So it's important in this big picture that we've been talking about in terms of regulation that cells be able to control enzyme activity. If they don't control enzyme activity, then they've got real problems. Whether it's an enzyme that breaks down proteins, or it's an enzyme that breaks down fat, or as we'll see later in the term, enzymes that break down glycogen and glucose. Being able to control these is a very important thing, and the different ways in which cells do that are important for us to understand. Yes, sir? Oh, yeah, that crazy damn thing. There we go. Okay. All right. Questions about the scheme? Yes, back here. So as you can see, trypsin is a master controller of all of these down here. That's correct. Over here, you.
Okay. So in this, is the trypsin the more important controller that we want to worry about, or the enteropeptidase?
Well, I don't want to say more. I mean, this obviously controls 4 of these guys, and we'll see trypsin plays roles with other enzymes as well, and enteropeptidase controls that. So there's just a hierarchy of these is really all that there is. Do you have a question?
Yeah, do these other molecules follow a similar scheme where they started with a single long protein chain, which is disulfide cross-linked, and then selectively cleaved to be activated?
Okay, his question is, do the other enzymes have this phenomenon of getting chopped into pieces, hanging on to pieces, like with disulfide bonds, and then becoming activated? And the answer is there's a wide variety of schemes that are used. In some cases, loss of a piece is important. In some cases, simply structural change is important. So there's no one answer to that question. And you had a question back here?
With the arrow going back from trypsin back to its
Yep.
right there, how does that work? Does trypsin also regulate its
Okay, good eyes. You have good eyes. All right. So as the figure implies, trypsin actually does play a role in helping to activate itself, and it's a complicated process. So trypsin can, once you've made some active trypsin, trypsin can then act on trypsinogen, kind of like we saw with chymotrypsinogen becoming partly activated. So yes, that can happen. Okay. So that's cool. Now, I said I was going to tell you something else about elastase, and this is a really cool story. All right. So let me tell you this story. What's up? Question? Question? Oh, yeah. Okay.
So the trypsin doesn't pull, like it can't, it seems like if trypsin can activate itself, as soon as you start one, it's never going to be inactive.
Yeah. So as I said, it's a complicated process, as trypsin can activate itself to some extent. A lot of this is going to be controlled by when and where that activation occurs. In the digestive system, you want to be able to activate as much as you can very quickly. So if you don't activate any of it until it gets to the digestive system, that is, you don't have any the master one, I can't remember the name of it, but the master enzyme, if you don't have any of that present where trypsin is being made, you're okay. But as you could imagine, you could see things kind of go haywire. Maybe things do back up a little bit. Maybe you're activating trypsin where you don't want it to be activated, and that's what's going to be a mechanism for making pancreatitis. Okay? Connie?
For the proteins that you just showed, do they all digest proteins except for lipase, which digests nothing?
Okay. So let me make sure I understand and answer your question properly. So if I talk about this, all the enzymes here are proteins. All the enzymes from here over to here digest protein down into other proteins, and this one simply breaks down lipids. Okay, in this case, fats. Okay? But all these, including this, is a protein. Right, yeah. Okay, yeah.
Produced to regulate themselves, say you have excess lipase or something, but you don't want to shut down all of them. They would have some kind of inhibition response further up their own chain that would maybe say get rid of the pro-lipase or something so that you didn't interfere with the other ones? Or?
I'm not sure I understand your question. Are you saying that if you'd want to be able to activate only this one and not these guys? Is that what you're...
Something like that, or maybe you just want to only...
Yeah, it's, as you can imagine, a fairly complex system. And in the context of our digestive system, we basically want all these guys to be active. Okay? We want them there breaking stuff down. So it would be unusual. We would have a situation where we would want to make this one be active and not these. And as you can see, it would be hard to do that because we've got one master enzyme that's catalyzing. Does that make sense? Okay, good questions. Glad to see you guys are thinking about this stuff. All right, I want to tell you my story. This story is a very cool one. All right, this story has to do with what you see on the screen here, and it's something called trypsin inhibitor. More specifically, it's called alpha-1 antitrypsin. What is alpha-1 antitrypsin? Alpha-1 antitrypsin is a protein that acts kind of like that regulatory protein of protein kinase A. It will sit on trypsin in the active site and prevent trypsin from breaking other proteins down. Alpha-1 antitrypsin is another way of controlling trypsin. So your question back here about if I have trypsin active and I don't want it to be active, do I have a problem? Yes, you could have a problem. But if you have alpha-1 antitrypsin present, the body has another way of controlling trypsin once it's been activated. Okay? Once it's been activated, you have antitrypsin, alpha-1 antitrypsin that combine to it, and you're set. Okay? Well, it turns out that alpha-1 antitrypsin is misnamed. It was named because the first enzyme that people discovered that it worked on was trypsin. But it turns out it works on some of the other proteases as well. And the one that it works on really well is elastase. Some people say, in fact, that it's better named as anti-elastase instead of alpha-1 antitrypsin because it's really good at knocking down the activity of elastase. And this is where the story gets interesting. Elastase is an enzyme that breaks down proteins, and it's present not only in our digestive systems, but it's also present in our lungs. Why do we have a proteolytic enzyme present in our lungs? Well, think about what our lungs are exposed to all the time. We're continually breathing in crap from the air. That crap from the air includes bacteria and viruses and all kinds of things. Our lungs are an interface between the rest of the world and our bodies. Having some sort of a protection against those things coming in is very good. Our lungs have elastase in them to break those guys down. They also have in the lungs alpha-1 antitrypsin. Now, why do they have both? Well, it turns out that just like we worried about having too much trypsin be active, so too do we want to control how much alpha-1 antitrypsin, how much elastase we have active. Because if we have too much elastase active, the elastase starts doing something we don't want it to do, which is attack our lung tissue, and that creates emphysema. So having the proper balance of alpha-1 antitrypsin in our lungs that's functional is very, very important. If we don't have enough alpha-1 antitrypsin in our lungs, we will develop emphysema. If we have too much, we may be more susceptible to infections. Balance is good. Balance is important. What disturbs that balance? Well, it turns out smoking disturbs that balance. Smoking oxidizes a very critical methionine side chain in alpha-1 antitrypsin. When it oxidizes that side chain, we create the side chain that looks like this. This is what we started with over here. Remember how I said alpha-1 antitrypsin fits into the active site and blocks it? This guy doesn't fit in the active site anymore. The more you smoke, the more you damage your alpha-1 antitrypsin, the more elastase starts doing the thing that it's supposed to, which is attack proteins, but those proteins are unfortunately in your lung tissue, and you're much more likely to develop emphysema as a result of smoking. Okay? Very nasty thing. Many of you who know me know how anti-smoking I am. Okay? With good reason. This stuff is designed to kill you. Yes, sir?
So is the protein that elastase attacks, I'm assuming elastin or something along those lines, is that present in large capacity in like the alveoli? And that's what leads to the alveoli collapse of the emphysema?
His question is does elastase preferentially attack a protein in the alveoli? And I don't know the answer to that question. I don't think it's preferential, no. I think it's a byproduct of that. But it is a concern to consideration. It's the alveoli that are affected by emphysema. But I don't know if it's specific, if it's really targeted there or not. Okay. You guys ready for a song?
Oh, yeah.
I just happen to have one. It's actually relevant to this, and it's a song. I'm not going to sing it. We're actually going to. I've got somebody who's recorded it for me. It's on YouTube, and it's called "I Lost a Lung." Okay, I got to set it up here, and here we go. Here's the. If you want to sing along, you can sing. Whoa, whoa. I'm not done yet, in case you guys are interested.
I'll not forget how you took my breath away. I lost a lung from smoking candles. Emphysema again. It seems to me you see those nicotines and tars leave alveoli
Okay. Let's see. This and this. Okay. So that's my little sermon of the day, I suppose. I've got about 5 minutes, and so I want to finish up talking, or at least introducing, one last topic that I'll finish up next time. And that's another important process that's controlled zymogens. Okay? Now, this process turns out to be absolutely critical for us to function because it controls our blood clotting. And again, it's kind of like our blood clotting is kind of like our digestion. We don't think about it, but by golly, where are we without it? Let's think of the challenge that our body has. Our body wants to deliver blood to our tissues, and it wants to repair itself if there's damage. So if I poke a hole in one of my arteries or one of my veins, I want to be able to stop that flow from happening. And not only do I want that flow to stop from happening, I want the repair to be done in minutes. Because if I don't, I will lose all of my blood. So I need to have a system that is extraordinarily rapid. And the way that I make it be rapid is I load my bloodstream with zymogens that are capable of forming that clot. So we can think of our bloodstream as kind of like a loaded gun. A loaded gun, because it's ready to clot as soon as the sense of damage occurs. Can we see problems happening with that? You betcha. If we clot when we don't want it to clot, we may be blocking blood flow to a critical tissue like our heart or our brain, and we have a stroke. If we don't make that clot, let's say that we are a hemophiliac, we will bleed ourselves to death. So again, balance is important. We have to be able to stop that blood flow and stop it in minutes. And this is the most amazing thing to me. It's got to be watertight. We have to assemble this block from molecular pieces. They have to be self-assembling, and it has to be tight enough to hold water. Okay? I'll stop with that, and that's where we'll pick it up next time. But I want you to think about that. Yes? Can pH activate zymogens? Generally, no. No.
Can
Yes,
I
sir.
give you this?
Would you mind bringing it back? I'm always afraid of losing them on the way back. So if you just bring it back to my office. If I'm not there, you can leave it with the secretary in the main office. Okay?
Sorry, I have
Yeah.
a couple questions concerning
Okay.
grading, so
Yeah.
maybe you need it.
No, please bring it to my office.
Where is the office, sorry?
Okay, I'm in ALS 2145. If I'm not there, you can leave it with the secretary in the. Okay.
I was wondering if you might be trying to talk about the bitumen.
Sure. Take a look at my calendar, and you'll see my calendar's pretty busy. But if you squeeze in between things, I'll be happy to talk to you. Okay. So just come by my office.
To discuss it.
I'm sorry?
Is today the last day to discuss it?
No, you can come today or tomorrow. That'll be fine.
Today or tomorrow?
Okay. But just come and wait, and I'll squeeze you in. Okay.
Okay.
Okay. How you doing, Veronica?